Let's now look at photosynthesis and the light-dependent reactions. So chlorophyll and accessory pigments. Chlorophyll A transfers light energy for carbon fixation. So in the light reactions that we're going to be talking about, chlorophyll A is what transfers that light energy to eventually be used in carbon fixation, which is the beginning of the light-independent reactions of the Calvin cycle. Chlorophyll B and the carotenoids act as accessory pigments absorbing wavelengths of light that chlorophyll A absorbs poorly. Okay, so these help maximize the absorption capacity of plants by capturing the different spectrums that chlorophyll A itself cannot capture. An example of carotenoid pigments includes beta carotene. There's two hydrocarbon rings connected by a hydrocarbon chain. When we're talking about beta carotene, you can see that on the left side there. And you can see that structure has alternating single and double bonds throughout that hydrocarbon chain there. Like the porphin ring in chlorophyll that we saw in the previous lesson, electrons in this chain of hydrocarbons in beta carotene are able to absorb light energy in the 400 to 500 nanometer range. Okay, so we see that this is a different range than the chlorophyll A's. Carotenoids reflect photons, those packets of light, in the yellow to red range and appear this color to the naked eye. So whatever the accessory pigment or the pigment itself cannot absorb, that's what's reflected, and that's the color that we see with our naked eye. So let's think of regular leaves, for example, in the summertime. Most often they are green. And it's not because chlorophyll itself is green. Oftentimes we color it that to signify, okay, green leaves, but it's actually not that case. Green plants that we see, that green is reflected off back into our eyes because that green is not absorbed from the light spectrum. Okay, so it might absorb the reds or the violets and blues, and the greens are not absorbed by certain pigments, and that would allow for it to reflect back and be seen as green to us.
So let's talk about photosynthesis. Photosynthesis can be divided into the light-dependent reactions and the light-independent reactions. Light-dependent reactions, often just called light reactions. It involves the direct use of sunlight, and this is where energy conversion reactions are occurring. We're taking this solar energy and we're converting it to chemical energy. In terms of photosynthesis, we're going to be seeing those high energy molecules such as ATP, just like in cellular respiration. We're also going to be seeing something very similar to cellular respiration, just a slightly different form. Instead of NADH that we see as a high energy molecule in cellular respiration, we see NADPH being formed through photosynthesis, which is a high energy molecule that we will be seeing through the light reaction. Then we have the Calvin cycle, which is a light-independent reaction. It does not require light to continue forward. We call this the stage 3 of photosynthesis. And this is where sugar is built, that glucose molecule. So it uses that energy that is harnessed during the light reactions, that ATP and the NADPH, to reduce the carbon dioxide that comes in through the stoma of the leaves. And this is where the synthesis of glucose occurs.
During the light reactions, we are going to be seeing an electron transport chain again. Okay, when we talk about the synthesis of ATP, we're going to be again seeing ATP synthase, that enzyme, and we're going to be seeing it embedded in a membrane once again with electron transport carriers that create a hydrogen gradient that eventually drives the synthesis of ATP, just like cellular respiration in the mitochondria. There are a few differences, which I'm going to highlight in a moment, but here is a picture of the mitochondria on the left. Okay, comparing it to the chloroplast on the right. And when we're talking about the chloroplasts, specifically, it's the thylakoid, those little discs that make up the grana, okay, that we're going to be seeing the electron transport chain embedded in the thylakoid. It'll be right in the thylakoid membrane there. And we'll be looking more at the similarities and differences in a moment. So the light-dependent phase, as I mentioned, occurs in the grana. The sunlight energy hits chlorophyll and will excite electrons. The return of electrons to ground state provides energy for two processes. So we're going to be seeing something called photolysis. Again, there's that word lysis, splitting. Okay, photo meaning light. So we're going to be using light to split something. What is that something? We'll be talking about that in a moment. Phosphorylation. We're going to be seeing the formation of ATP once again through that electron transport chain that was discussed. So photolysis, what is actually being split during the light reactions within the thylakoid? So we're going to be seeing a splitting of water into its main constituents, which is oxygen gas and hydrogen, as well as energy. Okay, so we'll see this happening, and we'll talk about the reason why it happens and why it's beneficial. But oxygen is the waste product, although it's seen as the waste product in photosynthesis because the main idea of photosynthesis is to finally produce that glucose molecule, right, to drive the energy of cells. Well, oxygen is a really handy waste product or byproduct for any aerobic organism that requires oxygen. So although it's considered a waste in the actual equation, it really isn't. It's very beneficial. Photolysis also provides electrons that will be excited when light is absorbed, and it fuels ATP formation, which we'll be talking about in photosystems 2 and 1. Hydrogen excess drives the rest of the reaction. So oxygen is that waste product, and then the other part, the hydrogen that splits from the water, actually drives the rest of the reaction. Okay, extra hydrogen picked up by hydrogen carriers. We'll see NADP being reduced and picking up that extra hydrogen into NADPH2. And this high energy molecule diffuses out into the stroma for use in that light independent phase during the Calvin cycle when light is not necessarily required. Lastly, let's talk about phosphorylation for a little bit. So the formation of ATP through an electron transport chain and the production of a hydrogen gradient across the thylakoid membrane involves phosphorylation. We have ATP that is used in the dark phase, or the Calvin cycle, or we call it the light independent phase. And we also see the formation of NADPH from NADP through redox reactions. So again, we'll see phosphorylation happening very similarly to the cellular respiration. So let's look back at this overview image once again. We can see that the light reactions are occurring in the thylakoid there. We see that light is being harvested into the chlorophyll that are embedded in the upper membrane of the thylakoid. We see that water is required. And as a product of splitting, we see that O2 is being removed from the system. It's being released. Okay, so we see that happening in the light reactions. We see from the light reaction, ATP is going to be produced, which will be utilized in the Calvin cycle. We also see that NADPH, that high energy molecule for photosynthesis, will also be released as a result of the light reactions, which will be used in the Calvin cycle as well. Welcome to the electron transport chain of photosynthesis. This is happening in the chloroplasts, specifically in the thylakoid there. You can see on the diagram, it shows thylakoid space. This is the area that transforms light energy into chemical energy of ATP. An electron transport chain requires NADH to donate electrons to start the electron transport chain. In this case, it uses the high-energy molecule, NADPH. The flow of electrons is triggered by light in this case because it's photosynthesis. NADPH is going to be one of the products at the end of the electron transport chain. So going back to the idea of light, since this is photosynthesis, there you can actually see two parts in the electron transport chain that light is coming in. Light will first hit photosystem 2, and then you can hitting photosystem I. These photosystems are named based on the order in which they were discovered, not the order in which they function. So remember, photosynthesis is endergonic. It's not exergonic, where it's releasing energy. Endergonic, it's taking it in. So in this case, the electron transport chain is driven by that light energy that's coming in. We'll look at this a lot more in depth in the next few slides. So here's a diagram of stage 1 of photosynthesis, which is photoexcitation. And again, this is when the absorption of light photons is occurring by an electron of chlorophyll. So we could see a chlorophyll molecule there. We could see photons coming in, and we can see that electrons are increased in energy because of this incoming light. And we say that they're excited. There's excitation. These excited electrons is captured by the primary electron acceptor, which oxidizes chlorophyll and reduces the primary acceptor. Okay, so that electron moves and is captured by that primary acceptor. If it's not taken by the primary acceptor, that energy has to go somewhere. So the excited electron falls back to the ground state and releases actually bits of light and heat. And this is the phenomenon of fluorescence. You could see in this diagram below, it is a reef that has photosynthetic cell. And in some cases, those electrons are not accepted and they release a glow. So complex of chlorophyll molecules, accessory pigments, and proteins are embedded in the thylakoid membrane, as we spoke about. And there's actually something called an antenna complex, which consists of several hundred pigment molecules. And this antenna complex absorbs photon energy and transmits it from molecule to molecule. So you could see that these antenna pigments absorb and it kind of bounces from molecule to molecule. This process continues until it causes the oxidation of chlorophyll molecule in the reaction center and reduces that primary electron carrier there. There's photoexcitation. The electron gets excited, bounces and bounces and bounces until it's finally accepted by the primary electron carrier there. And the primary electron carrier is part of the reaction center. So we're going to be speaking about photosystem 1 and 2, or more accurately, we should probably be saying 2 and 1. These two types of photosystems work together. They don't combat each other. They actually work together to be able to absorb different wavelengths to maximize the energy absorbed or taken in during this process. So photosystem 1, which actually appears second in the electron transport chain, sometimes it's called P700 as it absorbs wavelengths within the 700 nanometer range, which is red. That's the far right part of that visible light spectrum. And then photosystem 2 is known as P680. Again, the 680 tells us that it absorbs wavelengths in that area, which is more of like the orangey red. And the proteins associated with each photosystem determines the different wavelengths of the light that each can absorb. Okay, so let's talk about these photosystems. So this is stage 2, which encompasses photosystems 2 and 1. And this is part of the electron transport chain and chemiosmosis. So we can see that photosystem 1 comes second in the chain. And it's actually, in fact, photosystem 2 that is shown first. So photosystem 2, we know that it absorbs light in the 680 nanometer range. This is the first site of photoexcitement of electrons due to incoming light. And then we see photosystem 2 a bit further down the electron transport chain. And you can see that, again, it captures more sunlight. And these work together to absorb maximum amounts of sunlight. So let's look at all of the details of photosystem 2 and then photosystem 1 that is found along the electron transport chain in the thylakoid membrane of the chloroplast. This is depicted a little differently. You can see it's kind of on a y-axis of sort. It's not a very accurate graph, but it's kind of showing the steps in relation to how much energy is in the electrons. When we're talking about photosystem 2 and photosystem 1, there's kind of 6 generalized steps that occur for ATP and then the final high energy molecule to be produced. So we can see over here, this is stage number 1 or step number 1 in photosystem 2. And you can see that photosystem 2 is starting to absorb some light. We see sunlight coming in there. And this excites electrons, which passes from the chlorophyll to the primary electron acceptor. Okay, eventually that electron will need to be replaced. We'll get to that soon, okay, with more photoexcitement. But an enzyme actually extracts electrons from water. Remember, water has to be input into the system, and it supplies them to the chlorophyll to then be regenerated because those electrons were kind of lost up to the electron acceptor. So this splitting or photolysis that we talked about before is actually through that process, it's splitting H2O into oxygen gas, which is one of the so-called waste products of photosynthesis. It's contributing hydrogen, and it's also contributing electrons to replace the electrons that rose in excitement because of energy from the sun, in which case those new electrons can then eventually rise up to that primary acceptor as those original electrons start moving down the chain. Okay, so here is step 2. You can see it circled in that blue box. And this is where photolysis occurs. Okay, so there's going to be a splitting of water. We could see the diagram up above that water is one of the reactants of photosynthesis, and this is where it comes into play. So water actually splits through the help of an enzyme, and H2O is going to be split into 2 hydrogens and 1 oxygen. And at the same time, electrons are going to be lost in this process. And where those electrons go, you can see the splitting on the diagram on the right. It's showing plant splits water. And those little red electrons are actually going to go and fill the vacancy of where the chlorophyll lost its original electrons due to photoexcitation. So this is how electrons continue to be replaced and move to a higher energy state as more light comes in, and it generates a huge Poisson gradient across the membrane as electrons flow through the electron transport chain to eventually create ATP. So let's keep looking at how this happened. Here we have steps 3 and 4. So those original electrons that had photoexcitation due to the incoming light that was originally accepted by that primary acceptor, we could see that starting to move across. It's showing the cytochrome complex there in step 3. These are the complexes of proteins embedded in the thylakoid membrane that enabled electron transfer chain to occur. So you could see those electrons starting to pass along the complexes, and as the electrons pass through the complexes, this activates those carrier proteins to pump hydrogen protons across the membrane. And this is what's going to enable that gradient to form, just like in cellular respiration electron transport chain. This electron flow provides energy for chemiosmotic synthesis, as you can see in step 4, for that ATP synthase enzyme at the very end of the electron transport chain to conduct chemiosmosis and convert ATP through phosphorylation to ATP, AVP to ATP. And we can see that ATP is produced due to the electron transport chain and chemiosmosis, in this case for photosynthesis. So when ATP is formed due to the input of light from the atmosphere, this is actually called photophosphorylation. And this is the light-dependent formation of ATP. So it's different than the phosphorylation that occurs in cellular respiration, because in this case, it's triggered and facilitated by light exciting those electrons to initiate the electron transport chain to eventually form ATP. You can see that the ATP then is going to be moving into the Calvin cycle next to be used to build those carbohydrates, to build the glucose that is the end product of photosynthesis. So we're not done yet. You would think we are because, okay, we've made ATP. That's all we need, right? But we also need those high energy molecules for the Calvin cycle as well. So we're going to now move into photosystem 1. So photosystem 1, you can see more light is coming in, and that light is going to stimulate the chlorophyll and excite the electrons in the chlorophyll. And you can see those electrons in photosystem 1 are now going to move up to an excited state and also be accepted by a primary acceptor, just the same as in photosystem 2. But this is just a different wavelength of visible light that's being absorbed in this case, right? Here, these excited electrons are going to continue passing through the final stages of the electron transport chain. Okay, this is not the end. The photosystem 1 is kind of like a halfway point along the electron transport chain. So these electrons will continue to move across, and instead of them contributing to a gradient that produces ATP in the end, it's going to move along and actually pass through and activate an ATP reductase, the enzyme embedded in the thylakoid membrane. And because of this activation, this is going to reduce NADP that is found in the thylakoid, and it's going to create that high energy molecule NADPH. Okay, and this is the high energy molecule that photosynthesis creates in the light reaction, and the Calvin cycle, which is the light independent reaction, depends on to carry out its function. So you can see the little piggy bank there at the bottom. It's to remind you that in photosynthesis, the money in the bank is the NADPH in that case. That gives us that high energy molecule. So here's a big review of the overall electron transport that's happening in photosynthesis, that electron transport chain. Okay, again, you see photosystem 2 coming into play first. We see that there's photolysis happening, okay, and this is what creates that oxygen gas as one of the waste products. We also see the movements of electrons because of the sun, because of photoexcitation. It comes across those protein channels. It's creating gradient that will eventually be used to photophosphorylate the ADP to ATP. And then photosystem 1 will come in, absorb more light as energy, and photoexcite more electrons of those chlorophyll, and those specifically go to make NADPH, that high energy molecule. And now we have both ATP and NADPH moving into the Calvin cycle. Okay, and you can see that the Calvin cycle, that those high energy molecules in the ATP are moving out of the thylakoid, right, which are those discs which make up the stacks of grana, as you can see on that diagram there. And it's going to be moving into the stroma, which is kind of that fluid-filled space inside the chloroplast for that light-independent reaction of the Calvin cycle. So just to recap the electron transport chain of photosynthesis, It uses light energy to produce APP and NADPH. And these two are precursors and necessary to move into the Calvin cycle. Photosystem 2 absorbs light. It excites electrons in the chlorophyll, and it moves it to the primary electron acceptor within the electron transport chain. And then they need to replace the electron in the chlorophyll, okay, by photoexciting, by splitting the water by photolysis, and oxygen is released by photolysis, okay, and then there's extra electrons when this is split due to an enzyme activating the reaction that splits them, and those extra electrons now come back into the place of where those electrons were lost from the chlorophyll, and that continues to be moving to the primary electron acceptor and down the electron transport chain to further increase that gradient to produce ATP. And then in the end, we end up having more oxygen as well being released, and we can breathe easier.